Hi, my name is Geppert. I'm a PhD student at the Karl von Ossietzky University of Oldenburg in Germany. I want to tell you about some of my recent research in the working group Systematics in Evolutionary Biology on nanoparticle consumption in microfauna and why this topic is an ever-pressing concern. Although most media attention has focused on the negative effects of microplastic in the environment, the recent years have also seen large increases of micro and nanoparticle pollution deriving from electronics or cosmetic industries or medical applications like cancer detection. But maybe I can describe this extent of pollution a little bit better. You've heard about microplastic? These are microscale beads or pellets that are specially manufactured and used for larger items. And the amounts of the so-called primary particles that are being used are staggering. So, for example, more than 4,000 tons of primary microbeads were used just for cosmetics, just in Europe in 2012 alone. So, if we assume that one microbead has a diameter of 1 micrometer and an average density of 1 gram per cubic centimeter, then those 4,000 tons correspond to 4 by 10 to the power of 21 worth individual particles. To put this number into context, it is 1,000 times more than estimated number of sand grains on all beaches on Earth. Or a more available number to comparison is the number of the inhabitants of Germany, which is nearly about 80 million. This is 100 billion times smaller than the estimated sand grains, but it doesn't stop there. Particle pollution can also arise through the abrasion and disintegration of these primary particles or other waste items, resulting in increasingly smaller so-called secondary particles, many of which are about 1,000 times smaller and so exist in the nanoscale. Particles on this scale, which also includes viruses, are so small that they can only be seen with an electron microscope. Through industrial abrasion processes, simple waste, sewage, or diffusion into groundwater, much of the particle pollution ends up in the aquatic environment, where it affects all organisms from the very large down to the invisible and mostly unknown. An example of the latter are the microscopic rotifers, one of which you can see here. The individual of the freshwater species Brachionus calyciflorus is wheeling in some algae, feeding on them, as well as on many other particles suspended in this water column. Rotifers are not the only group of organisms to comprise the microfauna community in aquatic habitats, but they are an important one and occur worldwide in both freshwater and marine environments, as well as in brackish environments between these. They are also one of the first steps in a long food chain that eventually leads to us. To investigate the possible consumption of the nanoparticles by the rotifer Brachionus calyciflorus, our investigations started with our colleagues in the working group of physical chemistry of Professor Dr. Al-Shamari here at the Karl von Ossietzky University in Oldenburg, who synthesized spherical citrate-stabilized gold nanoparticles with different sizes in a controlled manner for us. For instance, here we see gold nanoparticles with a gold concentration of 82 mg per liter and a diameter of about 12.5 nanometers. Although this might seem an odd choice of nanoparticle to use, gold nanoparticles are commonly used as catalysts in numerous chemical reactions, including many used in biomedical applications. After exposing our rotifers to the nanoparticles, the first results were already visible under a normal light microscope. Compared to the control individual without nanoparticles on the left, the one exposed about 8 mg per liter nanoparticles on the right shows dark agglomerations in the gastrointestinal tract. Additionally, a 6 times higher concentration was usually lethal and in as little as only 1 day. To get a clue what these nanoparticles are doing to the rotifers, we have to go even smaller to reach the nanoscale. So to see what's happening inside of the rotifers, we have to make cross-sections with an ultrathin thickness of about 70 to 90 nanometers, which is 1,000 times smaller than a human hair. So after preparation and embedding dehydrated individuals, it is possible to cut the hardened resin and to make some cross-sections using a diamond knife in the ultramicrotome. Each individual rotifer of Brachionus calyciflorus can yield about 4,000 of these ultrathin cross-sections, which we scanned through and focused on the digestive tract using a transmission electron microscope. Through our experiments, we were able to show the first time that the gold nanoparticles, here with diameters of about 12.5 nanometers, were actually being internalized in the tissue of rotifer individuals of Brachionus calyciflorus. This only occurred in the cells lining the stomach, where some individual cross-sections suggesting that the uptake might occur via endocytosis, a common mechanism for the uptake of small particles by cells. Moreover, the gold nanoparticles were found to be highly concentrated within the vacuoles of the cells. These findings has 2 important results. First, the accumulation of the nanoparticles in the cells of the stomach of individuals of Brachionus calyciflorus seems to be lethal at higher concentrations. Second, and more globally, the accumulation might not stop with the rotifers. As important members of the microfauna community, the rotifers serve as food source for, among other animals, fish larvae, which in turn also serve as food source for other animals. As such, and as it was the case for the insecticide DDT in the last century, there's the possibility for the accumulation of nanoparticles up through the food chain via trophic transfer. Although the most media coverage and indeed the most research has focused on the effects of microplastic in the marine environment, we stress here that Brachionus calyciflorus is a freshwater species and such habitats are at equal risk from our man-made pollution. And don't forget that we are producing beaches on beaches of these small particles for our electronics, cosmetics or our medical tests every year. If you are interested in our research and want to know more about the outcome and the details of our exact findings, including different particle sizes and concentrations, have a look on our publication. Or write to me at our working group, Systematics in Evolutionary Biology. Here, I especially want to thank the Physical Chemistry group for their cooperation, as well as Professor Dr. Beninda Emmons, Professor Dr. Al-Shamari, and Dr. Alris for their advice and support. Bye.